So good evening, everyone, and a very warm welcome this sunny Monday evening to the Cambridge Society for the Application of Research. We have two great talks for you tonight. I'm afraid Mike Gregory, our president, can't be with us, so I'll be taking you through proceedings this evening. In a moment, we'll be hearing from Jenna DiRito about organ transplantation, and after her, we've got Professor Sir Colin Humphreys to talk about graphene and gallium nitride. Before then, I'll just go through a few quick things about questions and answers. So if you want to ask a question, and would love you to ask questions, you've got two ways of doing it. One way is to type your question into the Q&A box. Please don't use the chat, just the Q&A box, and I'll do my best to read out the questions as they come in and as faithfully to the question as I can. If, on the other hand, you're happy to be seen on screen, please put your hand up. You have a virtual hand that you can raise, and if you do that, we'll bring you in on video so you can ask your question in person. I would warn you, though, that we do record the talks, so if you're happy to be seen on YouTube, that would be great. We'd love you to ask your question in that way. Otherwise, you've got the typed Q&A to go for. So, as I said, first this evening, we've got our talk from Jenna DiRito. Jenna is one of our student award winners. So CSAR tries to support students by giving out a number of awards each year. They're for excellence in research, but not just in research that has a real world application. And Jenna's work is all about organ transplantation, and she's going to be talking to us from Yale. Over to you, Jenna.
Great, well, thank you so much for that introduction. Let me go ahead and share my screen and we'll get started. So today I'll be talking to all of you about revitalization of human kidneys for transplantation. And basically I'm coming at this talk from the perspective of a transplant scientist, which means that instead of doing basic science as you would in a standard pipeline, which is, spending years developing cell models and then testing the drugs that you've developed in cells into mouse models and trying to translate them into humans, which anyone who's done basic science research before knows that takes so much time and so much money and is usually not very successful. Well, in transplant, we have an alternate pipeline because there are a ton of organs that get discarded. And so instead of just letting these organs go to waste, my lab actually can take them and we can study, well, one, why were these organs discarded? Two, how can we make them better? And three, how can we not just make them better, but make them transplantable again? And so that's really what I'm going to be talking to you about today. So let's just take a step back and think about, well, why transplant? Where's the need here? And so these numbers are from the US, but they're mirrored in the UK and throughout the world. So in the US, there are currently 100,000 people waiting for a kidney transplant. And those are just the people that can get on the wait list. That's not everyone who actually has renal failure or is in need of a transplant. And even though there are so many people who are in need and waiting for a transplant, there are only slightly over 20,000 transplants performed every year. And because there's this great disparity in the number of people on the wait list and the number of people who can get transplanted, this ultimately results in about 13 people dying each day, which is tragic and something that we want to fix. And so one of the ways we think we can fix it is with these discarded kidneys. So even though there are 13 people dying each day, there are still about 10 kidneys that are being thrown away each day. So after a donor has been deceased, their kidneys are retrieved for transplantation. And for one reason or another, the surgeon determines that these kidneys cannot be used for transplant and they end up going in the bin. And like I said, instead of going in the bin, we actually use them to study and create new treatments for them. And one of the treatments that we like to use in kidney transplantation is actually targeted nanoparticle technology. So nanoparticles, if you've never heard of them before, a quick little overview, are biodegradable. They've been shown time and time again to be safe in patients. You can customize their size so they go to specific parts of the body. You can encapsulate hydrophobic drugs in the nanoparticles we create, and they release over a prolonged period of time. And not only do they have these great release profiles, they can also be targeted to specific areas in the body. Now, a lot of ways that people think targeting works is that a nanoparticle will go into the bloodstream and immediately find the site of interest, like a homing device. But that's not actually the way that targeting works at all. So if you put a targeted nanoparticle into someone's body, it'll enter the blood just like any other drug. And this means that it'll inevitably get taken out of circulation by the immune system. About 90% of it accumulates in the liver. And because it's circulating throughout the entire body, there is a whole potential of off-target effects, which is not great for the patient. So the question is, will targeted therapies ever reach their site of interest? And the answer to that is maybe, but only by chance. So one way that we combat this in transplantation is using a technique called normothermic machine perfusion. And so this is a video of an NMP circuit. And what we do is we plug in one part of the circuit to the artery of a deceased human kidney. So this is a human kidney right here. Plug in the vein to the other, and we have this catheter here that's connected to the ureter, which means we can get out any urine. So we see that blood is starting to pump. In a second, you'll also be able to see that urine is starting to drip out of the ureter. And so this is a way that we could start to really assess how this organ is performing outside of the body. So this is great. We can measure blood flow, urine, we can deliver really lots of therapeutic agents here, and it becomes such an exciting and unique system for targeted drug delivery. Because now, instead of delivering a drug to the whole patient and trying to treat one specific part, we're delivering drugs exactly to the kidney. So we don't have off-target effects. We don't have to worry about how the rest of the patient will react here. We're really just treating the problems that the kidney is having. So we've done it before. We've delivered nanoparticles, and I'm just going to walk you through this figure a little bit. So the white here is the vessels that we see in kidneys, and the blue is every individual cell. The red is a targeted nanoparticle, and green is non-targeted nanoparticles. So when we see areas of yellow, that means that for some reason or another, the targeting didn't work any better than just randomly delivering the drug in there. And we really wanted to figure out, well, why in some kidneys did this targeting work really well, where we have high areas of red, and then some kidneys, this just didn't work at all. So we find that when we, look using confocal microscopy at these areas where the red and the green nanoparticles overlap, we see that there are actually red blood cells that look like they're plugging up the vasculature here. And so we wanted to say, oh, maybe there's some type of clotting. So in a normal vessel, when you have a clot, you have red blood cells here that are overlaid with a fibrin mesh. And so this mesh is fibrin, and it holds the red blood cells and platelets together and really forms the clot. And so we were expecting that if we took a look using scanning electron microscopy or TEM we would see the same pattern. We would see this clotted mesh. But that's not what we saw at all. Instead, in the vessels, we found that the red blood cells were actually stacking up like coins, which was new to us and something we didn't expect in the slightest. And so this is actually known as the Rouleau effect. And this phenomenon has been documented in literature since the 1960s. And it's also known to really be driven by fibrinogen. So fibrinogen is a protein, the precursor to fibrin, the precursor to that mesh. It's just the non-activated form of fibrin, but it can be shown not to make that clotted mesh, but to actually link the red blood cells together and cause them to form those coin-like stacks. So we're really thinking that at this point, because we're seeing these coin-like stacks, that perhaps fibrinogen is the reason why these red blood cells are coming together and clotting up the microvasculature. And that's exactly what we found. So when we did a stain for fibrinogen, we saw that it is all within the vasculature. We've done immunofluorescent stains and histological stains. And so this is a glomerulus, which is the filtering structure of your kidney. And we see that in the glomerulus, they're totally plugged up. So there's a lot of fibrinogen overlaid with red blood cells here. So not great, because if there's no blood going through, number one, there's no nutrients and no oxygen being delivered to these places, so there could be more acute injury and more stress. And if we wanted to treat the organ, we couldn't do that either because no drugs can get through. So we really wanted to start to think about, well, where is this fibrinogen coming from? It must be originating in the donor, but it turns out that it's not. So before kidneys are transplanted, they're usually stored on ice. And we found that when these kidneys are stored on ice, this fibrinogen is building up in cell structures called the tubular epithelium. So right here, whenever you see this red outside of the vessels, which are white, or outside of this blue area in these tubular structures, that's where the fibrinogen is. And we interestingly found that the longer we keep these kidneys on ice, the more red or the more fibrinogen seem to accumulate, which is, a really interesting thing, because when we think of keeping kidneys on ice, we think that it's static storage. We think that, okay, it's frozen, it's a period of suspended animation, the kidneys go on ice, and then magically a couple hours later when they're transplanted, they're fine. But they're not, because there are a lot of dynamic processes happening when we have these kidneys stored and being prepared for transplantation. So we find that, okay, it's building up over this time. And then when we put it on this normothermic machine perfusion device, somehow this fibrinogen is releasing from the tubular epithelium. So all of that red signal that you saw before in here is no longer in the tubules. It's now translocated into the vessels and is causing this clotting. And we measure this too in the perfusate levels. We don't see fibrinogen before we put the kidney on, and then when we put the kidney on, it's released into the vasculature. So, another interesting point and why this is so clinically relevant is that we've not only seen this on the pump, so not just on normothermic machine perfusion, but we've also seen it in periods of real clinical transplant. So kidneys that were stored for a long period of time on ice were then transplanted, lose that red signal in these tubules, and that red signal where there is now clotting within the vasculature. So this is not only an important problem, but it's clinically relevant. And so one way we targeted these microvascular obstructions, these red blood cell stacks, is by using a combination of TPA and plasminogen. So these are just thrombolytic agents. TPA is clinically used to treat clots, and usually plasminogen is circulating in your blood, but we had to add that in here because we just use a washed red blood cell solution. So the good thing about TPA and plasminogen is that when it creates plasmin is not a very specific degrader of fibrin. It will also cleave fibrinogen. So we're able to see that when we have TPA and plasminogen, we're actually able to bust these clots. So at the start of NMP, like I've showed before, we had these really congested areas of the vessels where you see lots of fibrinogen accumulating with the red blood cells. And then when we have an hour of this TPA and plasminogen treatment, we see healthy single red blood cells, which are in yellow, circulating throughout the kidney and not getting congested anymore. So definitely a very promising treatment here. And this treatment has led to more stable perfusion. So this is the measure of resistance in the kidney. And when we add the treatment, the resistance doesn't spike, which has been a really key sign of a healthy kidney in our experience. We find that these kidneys are producing more urine. They have lower markers of injury, not only in their tissue levels, but also in their urine levels. And we're also able to finally have better targeted drug delivery, which is very exciting. So kind of taking you back to the beginning, we're not seeing all of these areas of yellow anymore where there's non-specific targeting. We see these high areas and high concentrations of red, showing that the nanoparticles have actually been able to target and overcome the non-specific accumulation. And what's, I think, the most promising part of this study is that was all the work that I did throughout my PhD. And now that I'm a postdoc, I'm actually working on translating this from bench to bedside. So we're in the, we've just received IRB approval in the States and are currently putting together the first NMP trial in the US, where we're adding in TPA and plasminogen to save long cold stored marginal kidneys and hopefully get them transplanted soon. So the first transplant should be taking place in about January, which is very exciting. So thank you to everyone who's helped me in this project, especially the donors, because without them, this work would not be possible. Thank you to the labs in Cambridge and Yale for funding this and for really seeing this vision through. Thank you so much, and I'm happy to take any questions.
Thank you, Jenna. That was absolutely fantastic. And the sort of application which is incredibly useful, but I think we would all hope not to come into contact with it.
Yeah, I hope so.
Now, I can see that John Cook has his hand up for a question. Let's bring John in.
Mute, and I can't see my video, but never mind. That was great, Jenna. You answered the first question I was going to ask right at the end when you said it was to be used clinically fairly soon. But my other question is, what do you know what causes the generation of the fibrinogen in the morbid kidney? Does it happen in a living kidney, or is there some process that continually sweeps it away, or what?
Yeah, so we have preliminary evidence that it's a downstream effect of HIF-1 alpha. So when these kidneys are being cut off from oxygen, HIF-1 alpha starts to signal, and this is, you know, an acute protein response that's saying there's something wrong with this kidney. This is not natural, because it is very unnatural for a kidney to be in a box of ice. So the kidney doesn't really know what to do. It's not used to that native state, and it doesn't react very well. But interestingly, we find that this happens in all kidneys when they're stored in ice to some degree. So it happens slower in young kidneys that tend to have good prognostic outcomes, but in older kidneys, so kidneys from 75, 80-year-old donors that also have comorbidities, fibrinogen spikes really fast. And those kidneys are at higher risk for not functioning after transplant.
Okay, thank you very much. Great talk.
Sure, thanks for your question.
So we've got one other question from Peter, which I think we can take, which is, how long are kidneys stored usually before transplant?
Yeah, so it's very variable throughout the world and throughout the country, depending on where you are. So the standard is for kidneys 24 hours, which seems like a lot of time, but really isn't when you try to get all of those logistics sorted. And in the marginal kidneys that we've studied, we find that at 24 hours is really when that fibrinogen starts to peak. In younger kidneys, it seems to not be so detrimental. But yeah, 24 hours, kidneys are usually stored prior to transplant. And it varies from organ to organ too. So if you're looking at heart transplantation or liver, you only get about 4 to 6 hours. So it's very variable, and we're trying to figure out how to extend the preservation times of all of these organs.
Very, very important work. Thank you, Jenna. That was a fantastic talk.
Thank you so much.
Thank you very much, and congratulations on your award.
Thank you.